Great. Thank you so much for the introduction, Andy, as well as for the invitation to speak with this group today. I am very excited to share some of this work from my postdoctoral work at Boston University. And a second reason I'm really excited to present with this group is that some of you may or may not know that I'm actually planning to relocate to Brisbane at the end of this year, or as soon as the borders allow. So this is a really nice opportunity to begin to make links as well with the speech science community in Australia. So before I get into the details of the talk, I would like to just tell you a little bit of information, I guess, about my background and my research program. So I'm originally from Ireland, and I completed my clinical training in speech pathology there before moving to Canada to pursue doctoral training with Dr. Yana Yunusova at the University of Toronto. My PhD work primarily focused on the speech movement disorder in Parkinson's disease and its rehabilitation using visual biofeedback. This work uncovered some important links between the speech movement disorder in Parkinson's disease and speech intelligibility, but it also raised some important questions about the role of sensory information in the speech disorder in this population. And so it was really these questions that led me to seek out a postdoctoral position with Dr. Frank Gunther at Boston University. So my research examines the neural control of speech production, and specifically I investigate how we use sensory information when we talk and how that changes in the presence of neurological disorders such as in Parkinson's, as I mentioned, as well as in individuals post-stroke. So much of my work is theoretically motivated by the Diva model of speech production. So this is a prominent model of speech motor control in our field and was developed by my postdoctoral mentor, Frank Gunther, in the 90s. And since then has been, refined and reiterated upon. And so that's kind of like, I guess, like the center of my work, but then I use a number of different methods, including behavioral methods, computational modeling, and neuroimaging to further uncover the sensory motor interactions that occur during speech production. Today's talk is really going to focus on the behavioral and the computational modeling aspects of this program. I'm going to begin with a background information about speech motor control, and then I'll present findings from two studies. The first is a behavioral study that examined feedback control mechanisms in voice production, and the second is a modeling study that looked at the interaction between feedforward and feedback control. If these motor control terms are not that familiar to you, that's completely fine. I'm going to review them in quite a bit of detail in the next few slides. So why is it that I care so much about sensory information? Well, speech production is a motor act that has sensory consequences. And this idea is really nicely illustrated in this figure that I've adapted from Murphy and colleagues. And so if we take a look at this figure, it really highlights the key outputs of speech production as the acoustic signal and facial movements. At the same time, the speaker is hearing the acoustics via auditory feedback, and they're detecting the facial movements via haptic feedback, or what I'm going to refer to in this presentation as somatosensory feedback. And so there is an ongoing mapping between this sensory feedback and the motor commands for speech production. This mapping is really critical, not only when we're first learning to talk as infants, but also in the ongoing monitoring of our speech production. So take, for an example, a scenario where you're meeting a friend for a coffee at a cafe. As you enter the cafe, the background noise around you gets a lot louder, because of the ongoing conversations or the noise from the coffee machine and so on. And in response to that, you automatically adjust your production to be louder and to be heard over the background noise. That adjustment relies on your auditory feedback system. We can use models to gain a better understanding of the processes that underlie speech production, and the Diva model is one example of a model of speech production. So the Diva model describes both the control theory of speech production, and it also describes the brain regions that are involved in the different processes. Today, I'm going to focus on the control theory side. I'm not going to get into the neuroanatomical specification. And so here I'm showing kind of the schematic of the control theory, and there are three main components. So we have the feed-forward controller, the auditory feedback controller, and the somatosensory feedback controller. Production of a speech sound begins with a readout of a set of learned targets that are sent from the speech sound map here on the left to these three controllers. And so when we say sound in this context, it could be a phoneme, it could be a syllable, it could be a word, just a sound or a sequence of sounds that are well learned. The auditory and somatosensory targets represent the desired states of the sensory systems for that current sound. Then the motor target is basically the motor program for the speech sound and specifies the sequence of articulatory movements for the current sound. The feed-forward controller then compares the motor target to the current configuration of the vocal tract. And it is going to generate a series of articulator velocities that is going to move the articulators from their current configuration into the configuration that they need to be in to produce the speech sound. As speech occurs, the auditory feedback controller compares the incoming auditory feedback to the auditory target. If it detects a mismatch between the intended target and the feedback, it will send a corrective command to the vocal tract to correct for that error. And then very similarly on the somatosensory side, this somatosensory feedback coming in, the somatosensory feedback controller compares that to the somatosensory target. If it detects an error, it will issue a corrective command to the vocal tract. If these errors are sustained over a period of time, a portion of that corrective command will also be added onto the feed-forward command for future productions. And in this way, this system is not only reliant on feedback control, but can also learn and adapt given changing demands. Experimentally, we can use a perturbation paradigm to probe the speech motor control system. So the perturbation paradigm is a very common paradigm that has been used both in the non-speech as well as the speech literature. And in this paradigm, sensory feedback is altered, and then we can examine how people compensate for that alteration. In the speech and voice world, these perturbations can be provided in the auditory domain or in the somatosensory domain. So we're going to kind of walk you through what that might look like in the auditory domain. And so this is an example of a pitch perturbation task. Typically, participants are equipped with a microphone and headphones. They produce a sound. That sound is recorded onto a laptop and then is processed in near real time so that that signal is shifted or perturbed in such a way that the person will then hear that perturbed feedback in the headphones. And this can be done we're talking about, maybe 20 to 30 milliseconds. So participants don't notice the delay in the feedback they're hearing. They just think that that's what they're producing. Specifically in this example, which I took from Carly Demopoulos and colleagues, the participant is sustaining a vowel sound, the pitch is being perturbed. So they're hearing that the pitch of their vowel at a higher level. And then what happens is that participants will start to lower their pitch so that what they're hearing in the headphones is closer to what they intended. And this paradigm can be used with other acoustic features as well. So it could be the speech formants, it could be loudness, it could be spectral features, and so on. An important classification in perturbation paradigms is whether studies examine reflexive responses or adaptive responses. So on the reflexive side, the perturbations are presented just on a small percent of trials. And so what this does is that it makes the perturbation unexpected and the participants are not anticipating or learning how to adjust for the perturbation. On these trials, we see a within trial correction, but because the perturbation is not sustained, we don't see any update to the feed forward command for the next trial. In contrast, in the adaptive paradigm, the perturbations are sustained over many trials, and we see both a within trial correction as well as an update to the feedforward command for the next trial. And so the reason I'm kind of bringing up this classification here is that the first study I present today is going to use the reflexive paradigm, and the second study is going to use the adaptive paradigm. So that brings me to the first study, which was a study of auditory and somatosensory feedback control in voice production. And so this was a study that was published last year in the Journal of Speech, Language and Hearing Research. And it was led by a recent PhD graduate in the lab, Dante Smith, who I co-mentored in the last couple of years of his program. So the majority of perturbation studies that have been done in voice motor control have been done in the auditory domain. So that's using that pitch perturbation paradigm that I just showed you. On the somatosensory side, only 2 prior studies have used laryngeal perturbations to displace the larynx during vocalization. In these studies, as they apply the perturbation, there was an immediate lowering in fundamental frequency. So that's the kind of the acoustic measure of pitch. And the authors in those studies also observed a compensatory response in the opposite direction. So it did seem that the perturbations in the somatosensory domain were eliciting a similar response to what we see in the auditory domain. But in these studies, when the perturbation was applied, participants would have both been able to feel that perturbation against the larynx and also hear the effect of the perturbation on the acoustics. And so it's not clear whether that compensatory response was being driven by the auditory or the somatosensory feedback controllers. So in this study, we aim to dissociate the roles of these 2 feedback controllers in response to laryngeal perturbations. And our key experimental manipulation here was to manipulate the availability of auditory feedback. So in one condition, the participants were able to hear their vocalizations. In the second condition, we played a loud white noise, which had the effect of masking their ability to hear themselves as they were vocalizing. We also had a second aim, which was to compare responses to these laryngeal perturbations to responses to auditory perturbations. So there were 18 native speakers of North American English who took part in this study. They were all between the ages of 18 and 35. They had no reported history of speech, language, hearing, or neurological disorders, and had no formal singing instruction or experience speaking a tonal language. The task was a sustained vowel production of the vowel E. On about 25% of trials, a perturbation was applied, and then at the end of each trial, the participants received visual feedback about how loud they were speaking. The idea here with the visual feedback was to keep them vocalizing at a relatively constant level or constant loudness throughout the experiment. And this is particularly important when you have a condition where you're playing loud noise, because people will automatically adjust to try and speak louder than the noise that you're playing in the headphones. The laryngeal perturbation consisted of a physical displacement of the larynx using a laryngeal displacement device so this is a lab-made device, and in the lab we refer to it as the Perturbatron. It consists of an inflatable balloon that is embedded within a collar placed over the laryngeal prominence, and then we can control like how and when that balloon is inflated using scripts in MATLAB. That inflates, it then pushes against the larynx, and the effect on fundamental frequency is that it lowers. And this is very similar to what the previous studies had done. The auditory perturbation was designed to mimic the effect of the laryngeal perturbation as closely as possible. And so this involved a 100-cent downward shift in fundamental frequency that was played through the headphones for the participants. So then in total, we had 3 conditions. I've kind of alluded to all of these, but I think it's just helpful to put them all together. So there's 2 laryngeal perturbation conditions. One where auditory feedback is masked, and that's masked using speech-shaped noise at 90 decibels. The second laryngeal perturbation is without auditory feedback masking, so they're able to hear their own vocalizations. And then the third one is the auditory perturbation condition. For each trial, the fundamental frequency is extracted using Pratt and then converted to sense so that we can compare the pitch traces across individuals. And so what you're seeing in this figure is an exemplary trace from an individual, and it has been time aligned to the onset of the perturbation. So 0 here is perturbation onset. Immediately after the perturbation onset, we see this lowering in fundamental frequency. So this is really the effect of the perturbation on the fundamental frequency. And then at around 100 milliseconds, or where this purple triangle is, we start to see a compensatory response where fundamental frequency starts to go back towards baseline. And so from this trace, we then extracted 3 different measures. The first was stimulus magnitude. So this is just estimating, what was the effect of the perturbation. The second is response latency. And so this is capturing how long it takes for a participant to start responding. And the third is response magnitude. So how much of the perturbation did they compensate for? In the laryngeal perturbation experiment, the stimulus magnitude did vary across individuals, so really just depending on how that equipment would interact with their anatomy. And so we did want to also calculate a normalized response magnitude measure that was normalized by the size of the perturbation. And so we had a 4th measure called response percentage, and that's just that normalized compensatory response. So each measure was submitted to a repeated measures ANOVA, examining the effect of condition on these measures. And so for the first 3 measures, there was no significant effect of condition, but for the last measure, the response percentage measure, we did see a significant effect of condition. So recall that this is the normalized measure of response magnitude. And so it's really getting at, what is the compensatory response to the perturbation? And we saw the largest response in the laryngeal perturbation condition without any auditory masking. So that's when they're able to hear their own vocalizations, followed by the laryngeal perturbation condition with auditory masking. And then finally, we saw the smallest response in the auditory perturbation condition. So looking at these results in just a little bit more detail, we saw the greatest compensation for laryngeal perturbations when auditory feedback was available compared to when it was masked. And so this suggests that both feedback mechanisms are contributing to the degree of compensation to a laryngeal perturbation, and that's why we see a greater compensation overall. It also suggests that the paradigm effectively dissociated between somatosensory and auditory feedback control mechanisms. And this really opens up the potential for using this paradigm for studying voice disorders, especially disorders where these feedback loops are implicated, such as in spasmodic dysphonia. We also observed smaller compensation for auditory perturbations, and this can be explained by the fact that during auditory perturbations, the auditory feedback controller detects an error, but the somatosensory feedback controller does not. And it's only as the auditory feedback controller begins to correct for the auditory error that the somatosensory feedback controller is going to detect an error. So I guess maybe just to put that in another way, the auditory feedback controller is correcting for the error. The somatosensory feedback controller is trying to keep the vocal tract in that baseline configuration. And so really it's this competition or the push-pull between the 2 feedback controllers that is potentially resulting in the lower compensatory response in this auditory perturbation condition. So overall, this study demonstrates how we can manipulate this perturbation paradigm to really get at that interaction between the 2 feedback controllers. Now in the next study, I'm going to show you how we take a different approach using modeling to examine the interaction between these 2 controllers, as well as the feed-forward controller as well. So this second study was also published last year. This is in Frontiers in Psychology. As I mentioned before, this study uses the adaptive perturbation paradigm. So I'm going to walk through this paradigm in a little bit more detail. So this is what a typical experiment looks like, with trial progression on the x-axis and the magnitude of perturbation on the y-axis. In this paradigm, it typically has 4 phases. So we see a baseline, ramp, holds, and after effect phase. During the baseline phase, participants will produce a word or sound or whatever stimulus is being investigated. They're going to, at this point, just hear normal auditory feedback in the headphones because the perturbation has not yet been applied. Then for each production, an acoustic measurement is taken. And so I'm just going to represent the acoustic measurements per trial with these red dots. Then moving on to the ramp phase, the perturbation is gradually and linearly applied. And in response to that perturbation, participants will begin to oppose it by moving their productions in the direction opposite the perturbation. Then in the whole phase, that perturbation is maintained, and kind of similarly, the compensatory response is maintained. And the last phase is the after effect phase, where the perturbation is removed. And what's interesting at this point is that typically what we see is that rather than the participants' productions going immediately back to their baseline level, we see this kind of residual effect in their productions of the perturbation. And this is really how I guess we know that there is some degree of learning occurring in this paradigm, in contrast to the previous paradigm where we don't see any residual effects over time. And so these responses really are like a combination of feed forward and feedback control mechanisms. But it's quite difficult to disentangle the contribution of each of the 3 controllers from the behavioral responses alone. And that's where modeling is going to come in. So the Diva model does provide us with a framework to think about how these interactions occur. But the computational implementation of the full Diva model actually has too many free parameters for us to be able to model these adaptive responses. And so what I mean by that is that there are many combinations of parameter values that will be able to be fit or could explain the same data set. And so it would not be possible for us to arrive at a unique solution or at a solution that we could really interpret. So the aim of this study was then to develop and test a simplified computational model that estimates the contribution of feedback and feed-forward control mechanisms to these adaptive responses. And so we've called this model the simple Diva model because it is still based on the Diva model, but just with a much more reduced set of parameters. The model is described using 3 equations, and these equations solve for gains in auditory feedback control, represented by alpha A, somatosensory feedback control, represented by alpha S, and feed-forward control, represented by lambda FF. I'm going to walk through the model equations based on an F1 perturbation example. So this is, in this example study, it is the first formant, the first speech formant that is being perturbed. And I'm going to bring back as well the schematic of the experiment just so that we can kind of just have a visual reference for what's going on. So in this example, we're going to say that there was a 30% perturbation applied, and so that's being applied to F1. And at this point, we're also going to assume that the single acoustic measure has been extracted from each trial. And if it's a perturbation being applied to F1, we would then also be measuring F1 in the acoustic signal. We're also going to make the assumption that the target value of F1 or F1T is equal to the mean of F1 during the baseline. So I guess the way to think about that is that the people's productions during that baseline phase are fairly representative of what they intend to produce because there is no perturbation. And so we're kind of just inferring then that this could be equal to their target value. And so again, just for the purpose of making things a little bit more concrete, I'm going to say that for this example, the target F1 is 500 hertz. So the first equation then states that F1 in a given trial is a combination of a feedforward command and a feedback-based correction if an error is detected. And so this information is very consistent with what I've told you before. We're now just formalizing it and putting it into an equation. So starting with the very first trial, the feedforward command is going to be equal to the target. And since there is no perturbation, there's no error detected. And so we can fill in what we know. We know that F1T is 500, and here there's no error because we don't have any perturbation. So the error for the very first trial is going to be 0. And this is actually going to be the same throughout the baseline trial. The model is going to estimate that baseline period as just a flat line. Once we do have a perturbation, that's where our second equation is going to come in. And so this is where we want to estimate what that feedback-based correction looks like. And so that here is equal to the size of the auditory and somatosensory errors, if there are any, and then scaled by gain parameters. So this is a little bit of a mouthful, but I am going to break it down further. So if we first begin with the auditory error part of the equation, sorry, I forgot there was a few extra animations. So the auditory error is estimated as the difference in between the target and the auditory feedback. And so in this example, the target is 500 hertz. And we also know what the auditory feedback is because this is something that we're actively manipulating as part of the experiment. So the auditory feedback is going to be equal to the feed-forward component as well as any perturbation that we're applying. In this case, it's a 30% perturbation. So then the auditory feedback component is going to be the 500 plus the 30% of that, which is 150. Then similarly, the somatosensory error is estimated as the difference between the F1 target and the somatosensory feedback. Since we're modeling auditory perturbations in this example, we're going to assume that the somatosensory feedback in this first perturbed trial is equal to the F1 value specified by the feedforward command. So for this first trial, we're not actually detecting any somatosensory error. And again, we know that this is 500 in this example. And so then we can start to fill in, all of the values that we know so far. We work these out. We discovered that we have an error of 150 hertz. Typically, however, people don't fully correct for the error that's detected. They only partially correct for it. And that's where alpha A is going to come in. So we call this a gain parameter. You can also think of it as a scaling factor. So let's say someone corrected for 50% of the error, then this alpha A is going to be 0.5. Then moving on to the final equation, this is the equation that specifies what happens to the feedforward command for the next trial. And so that command is going to be updated by adding a scaled version of the feedback-based corrective command. And so very similar to how the gain parameter works for feedback. Here, again, we don't see that full corrective command being added onto the feedforward command for the next trial. Instead, it's only a partial addition. And so the lambda FF is going to capture that. So again, if 50% of the corrective command was added on to the next trial, lambda FF would be 0.5. So in reality, we don't do this by hand. We don't work out trial by trial what this looks like. Instead, we stick it into an optimization routine and let that optimization routine decide what are the best parameters that describe a given data set. So when we put all of the equations together, there are three free parameters, as I said, that have to be solved for alpha A, alpha S, and lambda FF. And the way that we that I guess we think about these things is that a high alpha A would mean that there's a high or an increased compensation. A high alpha S leads to decreased compensation, and a high lambda FF indicates a higher learning rate or more adaptation across the experiment. So these model equations are then fit to a data set. We use an optimization routine to solve for the three free parameters. And the final optimized solution that is selected is based on the lowest root mean square error between the model fit and the data set. In our paper, we did include 6 data sets from 5 different labs. But in the interest of time today, I'm not going to go through all 6. I'm just going to show you a subset of 3 data sets. And all of these data sets used an F1 upward perturbation, like was in our example. All of the data were from cohorts of healthy adults, and the simulations were performed in MATLAB 2018A. So the first simulation modeled data from what I'd call a classic adaptation paradigm. So very similar to the example. These data were collected by Terry Scott at Boston University. And so she's kindly shared the data with us just for the purpose of modeling. The model figures that I'm going to show you are going to be very similar over the next few slides. So I'll walk through these ones a little slower than the later ones. On the x-axis, we have trial progression or block progression. The reason this is block and not trial is that we just took the mean over every 3 trials just to reduce some of the variability in the data. And then on the y-axis, we have that measure of F1. The blue is showing the actual data as well as the standard error across individuals. And then the red is showing the model fit. And so I guess there's a few things to point out about this figure. The first is maybe looking at the underlying data. And so the perturbation onset is here around block 20. And then as soon as the perturbation comes on, we see this lowering of F1 to compensate for the perturbation. And then once the perturbation is turned off, the F1 does start to go back towards the baseline values. But it's not an immediate thing, as I alluded to before. We do see this kind of residual learning where they do not automatically go back to baseline right away. Just from like a visual perspective, you would also notice that the model does seem to closely pattern with the data, and we do take a few different metrics to assess this fit. Today, I'm just going to share kind of a simple correlation between the model fit and the data, but we have also looked at, RMSE as well as some other I guess just ways to measure the fit. So in the right-hand side, then we have a correlation coefficient, which is 0.96. So that's a very strong correlation between the data and the model. And then we also have the optimized model parameters are shown both in the figure, and I've just included the text as well because I think it's just a little bit easier to read in the context of this presentation. So the way to interpret these parameters is an alpha A of 0.23 indicates that 23% of the auditory error was corrected for within trials. An alpha S of 0.17 indicates that 17% of the somatosensory error was corrected for within trials. And then finally, a lambda FF of 0.09 indicates that 9% of the corrective command was added on to the subsequent trials. So given that visually, the model closely approximates the data, the very high correlation coefficient and the unique parameter solutions that the model was able to arrive at, we would say that the model does provide an excellent fit to data from this classic adaptation paradigm. The next simulation then modeled data again from an upward F1 perturbation paradigm. And what's different about this study is that they also used masking noise on certain trials. And so these data were collected by Kiri Ballard at the University of Sydney. And again, the model plot is set up in the same way. The only thing that's different here is that these gray bars are used to indicate trials that had masking noise played during them. And so during these trials, because of the masking noise, participants were not hearing their auditory feedback. They're not able to detect auditory error. And what is actually really interesting, both in the model fit and in the data, is that even though there is no error here, and if their responses were completely reliant on auditory feedback, we'd really just expect their productions to stay in the same place. But we do see them start to return to their baseline values. And so it really must be the somatosensory feedback controller that is driving this response back towards baseline. And so we see this in the data, we see it in the model, and we also see it in the optimized model parameters, because you'll notice that for the alpha S, so this is the parameter describing the gain of the somatosensory feedback controller, here is 0.48, whereas in the previous paradigm that I shared with you, we only had an alpha S of 0.17. So in this example, where the somatosensory feedback controller seemed to be contributing more to the response across the experiment, that is also reflected in the model parameter. So then I just have one more set of simulations to share with you. And these ones were looking at testing the predictive ability of the model. So these data were collected by Sarah Ching Chow as well as Ayub Deliri at Arizona State. And in this experiment, the participants, again, it was the F1 upward perturbation, but this time they completed two different conditions. In the first one, there was a gradual ramp phase, and in the second condition, there was a sudden ramp phase. So instead of there being like a gradual and linear introduction of the perturbation, it just came on at like that full 30% value and then was held on for the rest of the whole phase before being removed. And so what we wanted to do here was first fit the model to data from one condition, extract these optimized parameters, and then use those parameters to fit the model to the second condition. When we use the model in this way, when you're fitting it to the second condition, the model is only seeing the pattern of the perturbation and the model parameters. We're not actually giving it the data at all. We're kind of showing it here so that you can see what that resulting model fit looks like compared to the data. But when we're actually running the model does not know what that data looks like. And so when we look at the correlation coefficients, for each of these simulations, we see again very high correlation coefficients in both the original model fit and when using the model to predict performance as well in the second condition. And this result did hold as well when we did it the opposite way. So we could first fit the sudden data and then predict the response in that gradual condition. So together, these simulations show that the model does provide excellent fits to these F1 data sets. In our paper, we also included fundamental frequency data sets, and similar there, the model also provided really good fits. These simulations also reveal some model properties. So specifically that the model is sensitive to the presence of masking noise, and that it's possible to predict group responses from one experimental condition to another, at least within the same group of participants. We're not yet sure if that would actually hold, for a different group of participants. Overall, Simple Diva can provide a mechanistic explanation for adaptive responses that are not readily interpretable from the behavioral data alone. As part of this study, we also released a Simple Diva app. So this is a cross-platform open source software. The model code is originally written in MATLAB, but we did compile the code in such a way that you don't need either a MATLAB license or you don't need to have MATLAB downloaded on your computer to be able to use the app. And so this is on the lab website. You can also follow the QR code here to find the software. And it can be kind of out of the box applied to data kind of as a research tool if you have this kind of data. I think it also has a lot of potential as a teaching tool for anyone who is studying speech motor control. So in the future, we plan to use Simple Diva to develop clear and testable hypotheses that can then be evaluated empirically, and also just to understand the differences in speech motor processing in individuals with communication disorders. We are also actively working to expand the functionality of Simple Diva. So one of our more immediate goals is to model reflexive data. So that's the data that, similar to what I showed in the first study. And we actually expect to be able to release that version of the model within the next 1 to 2 months. And we also want to be able to statistically compare model parameters between groups, because right now we can really only do that in a qualitative sense, kind of saying, yes, this group seems to have an alpha A that's higher than the other group, but we don't yet have a way to kind of statistically quantify that difference. So then just in summary, I've showed you how the interaction between the auditory and somatosensory feedback controllers can really depend on whether we're perturbing in the auditory domain or in the somatosensory domain. And I've also showed how we can quantify the contribution of the 3 different controllers to adaptive responses using the Simple Diva model. So with that, I would just like to acknowledge the funding sources that made all of this work possible. So that's the National Institute on Deafness and Other Communication Disorders, the National Institute on Drug Abuse, as well as the Australian Research Council. And then I must also acknowledge all of the work and support from my collaborators. This is by no means my work. It is work by a very large and dedicated team that includes members of the Gunther, Step, and Perturbation Labs at Boston University, Ayub Deliri and Sarah Ching Chow at Arizona State, Kiri Ballard at the University of Sydney, and Suan Chang at the University of Michigan. I'd also just like to thank you all for attending today, and I'm happy to take questions. I'm also happy to chat offline at another point, and you can always find me on my website or on Twitter as well. But thank you.
Thank you, Elaine. What an awesome talk. That was really cool. So we have about 15 minutes for questions. So I'll just open it up and feel free just to unmute yourself and ask away. Anybody? Okay, I'm happy to get things started. So one thing that struck me was the Perturbatron, cool name, is providing not only the movement of the larynx, but presumably also people would be quite aware of the extra pressure on their skin when it starts to inflate. And I feel like that's quite different to the auditory perturbation, where you'd probably take a little bit longer to click that the F1 or the F0 is moving. So I'm just in that result where you get less compensation for auditory than laryngeal on its own could actually, whether the laryngeal effect, I take your point about the push-pull thing. So when you've got the 2 fighting against each other, but I wonder if the greater compensation for laryngeal is actually something to do with having a more meta or salient kind of cue in the form of that extra skin pressure. Like that initial kind of like, oh, they're doing something to my neck.
Yeah, I think that's a very valid point. And definitely the laryngeal perturbation is not something that you're going to miss. Like it's happening and it's happening. For the auditory perturbations, it can vary. Some people know that they've happened. Other people can go through the whole experiment, and if you ask them in kind of like an exit questionnaire, they may not even know, right? So I think you do raise a really good point. I think one thing that would I guess lend support to the results is that the responses are starting to occur around the 100 millisecond mark. And so when we, in the past, when you look at these, responses in, auditory and somatosensory domains, I think that time frame is, within like what you would expect for those sensory systems. Whereas I think if it was a more cognitively mediated response, we would see a later response. And I think there has been some work that has shown, even just in auditory perturbation studies, where they've indicated that you might see an initial response that's more sensory mediated, but then there might be like a secondary response that is, a few hundred milliseconds later. So I do think that I think it's a valid concern, but that I think the speed of the response that we're seeing really indicates that it's something that's more sensory, motor based than like a cognitively mediated response.
Yeah, that makes sense. I guess one way to test that would be to allow people to have some kind of click or beep at the point of auditory perturbation at the start of that, and then that extra condition so that you could sort of have something parallel where they know, oh, now something's happening to the auditory feedback.
Right. Yeah. The other thing about that is with the laryngeal one, yes, you have this like quite, like certain, I guess, perturbation, but I don't know that everyone clicks that there's this physical thing happening, but also that there's this change in your acoustic output.
Right. Yeah.
So yeah, I think there's a little bit to it, but yeah, you definitely raise a good point.
I'm sorry, can you hear me?
Yes. Yeah.
Yeah, great. Yeah, I also had a question relating this Perturbatron machine. I was just wondering, so because you were telling that this sort of downward peak, the displacement sort of like frequency displacement is different for everyone because of anatomical differences, which makes sense, I guess. And you're sort of correcting for that afterwards in your analysis by normalizing it. But would it be possible as well, you think, to sort of tune it before the start of the experiment so that everyone's sort of showing the same magnitude? Like I'm just thinking of TMS where they do this sort of threshold stuff at the start of the experiment to have the same effect for everyone. Do you think something like that's possible? Or
I think it is definitely possible in theory, and I think also maybe in practice, but probably not with our current setup. I'm trying to think. So we tried to standardize, the placement, the size of the balloons, like all of these things are also lab made. But we really can only, and I guess like the pressure, like the force of the inflation, we also try and control for. But we don't really have control then over what that effect is going to have on their fundamental frequency. And so maybe it's something that we could work with, but I think it would be tricky, because I think you would have to look at, trying different placements, possibly trying different balloon sizes. I think there would be quite a bit to it, at least with the device that we have. I think there might be other, maybe other devices that have a greater degree of control where something like that would be possible. I know so these laryngeal perturbations are really not that common. There have been a few more studies that have done jaw perturbations where there is a force being applied to the jaw that, essentially, pushes it down, left, right, etc. And so in those studies, you can have much more, it's like a robotic arm. So it can be something like a 10 centimeter, like a very specified amount of like the magnitude, not necessarily the effect on the acoustics could still be quite different from person to person. But I think that kind of device or setup definitely offers more control than what we have for these laryngeal perturbation experiments. Yeah. And it's also, I think, like the larynx is just so much more complicated than the jaw,
Yeah.
And so just getting in there and applying something is quite challenging.
Yeah. Great.
I had a quick question about the F1 studies. Were these with a range of different vowels, or were they generally using E?
Most of them are actually using so words like bed, head, dead, Ted. Those are very, very common words. I think the Australian one did have hair, bear, care, that vowel.
Yeah, I was just wondering because the aggregated data looked like the F1 was decreasing like during the baseline phase, which suggested maybe some kind of like approaching a target or something like that. And I was just wondering whether it included if one's going in different directions, but yeah.
Yeah. And so if often the words are presented, so I guess like the most common way to set it up is that you have three different words, like let's say bed, ed, and Ted, and then And if they present those in blocks of 3, then we kind of just average over those 3 so that we don't actually see those. We're kind of just, we're not really, I guess, interested in the word effects. We're, you know, as such. So if it's set up in that way, we can just average over them. So then each block should really be fairly stable. In the fundamental frequency studies, we do see drift across the experiment. Like that is just something that is in kind of most fundamental frequency studies that we see, that just when you ask people to repeat the same word or stimulus over and over again, that their pitch actually does
decline, right?
You know, yeah. And so the way we deal with that then is have a separate, you know, run or block that has no perturbation. You know, we extract the identical measures and then subtract that away, so that you account for that drift throughout the experiment. We tend not to see the same. I guess maybe there is, you know, I would think some variation in the F1 studies, but it's nowhere near what we see in the fundamental frequency studies. And so for those, for the modeling, for the most part, we're looking at raw data.
Right, cool. Any more questions? I've got more. I can keep asking you.
I have one more. So I'm working on like speech from Parkinson's patients myself, and you said you also worked on that, and you were also interested in how this feed forward and feedback mechanisms work like in those populations. There's actually a colleague of me in University of Groningen who will start her PhD on exactly this topic. So that's quite interesting. I was just wondering what you would expect for people with Parkinson's disease, like which feedback mechanisms would be sort of damaged, for instance, or do you have any strong expectations there?
So far as I am aware, for the Parkinson's literature, most of the studies, the perturbation studies, have been done in the auditory domain, and they have been done both with F0 and F1. In terms of the details of the responses, I don't remember the differences offhand, but I do know that their response is different depending on whether it's F1 or F0. And in one, I believe their response is elevated relative to, you know, age-matched peers, whereas the other one, I believe it was like decreased. And so we do, you know, expect or know that there are like different neural underpinnings for, you know, that kind of segmental level control versus the prosodic level control, and that's possibly what's happening in those two perturbations. In our lab currently, we are setting up experiments to do both laryngeal and auditory perturbations, and so that will allow kind of I guess a test of the fuller system in a way. And we are combining that with fMRI so that we'll also have the neuroimaging data to see, you know, what areas are, you know, more or less activated compared to their peers.
I'm just thinking of, there are some papers from the 80s, I think, where they did some, well, it wasn't Emma, but like what we had before that. I think it's Logeman and Fisher, where they say something about there's less movement of the jaw, I think, and their argument for that is that there are less like muscle spindles that give you like proprioceptive feedback, which, well, basically somatosensory feedback. So in that sense, I think it's really interesting too that you can also like tease those apart and see what happens there.
Right, exactly. Yeah, because I think that the responses we're seeing for the auditory perturbations, like there is something very real there, but it's hard to interpret by itself in a way. So yeah, I think it will be interesting just to see how that plays out. But that's definitely a large study. I think it will take quite a while. We're just at the very early stages. So yeah, watch this space, but it's not going to be an immediate finding for sure. It's a pretty big undertaking as a study, but also in COVID and everything else. It's going to take some time. Yeah.
Great. Good luck.
Awesome. Well, we're coming up to 10 a.m. here, and it's definitely time to knock off work in Boston, I'm sure. Thank you very much. We're getting thanks in the chat as well. That was a really fascinating talk. So we really appreciate you tuning in, and it's exciting to hear that you're coming to Australia as well.
Yes, yeah. So hopefully we'll be crossing paths in person at some point as well.
Awesome. Thank you so much, Dr. Carney.
Thank you.